Code protein expression, a novel tool for plant virus detection. The protein expression and studies which plays a backbone of post-genomic era. There exist two types of protein expression system. First one is in vivo and the second one is in vitro. Protein expression which simply mean production of protein from a gene construct, and thus produced protein can later be used for production of vaccines and used in protein therapies like human insulin interferons, and in plant virology the recombinant protein produced can be used for virus indexing purposes. There are two types of protein expression system. First one is cell based system or in vivo system, in which expression host and expression vector plays an important role. We ligate our protein of interest into expression vector and transform the recombinant vector into an host and culture the host in a bacterial specific medium, later induce the host with the chemicals, and at last we purify our protein of interest from the expression host. As we have already discussed, in vivo method, two major components. First one is expression host. There are prokaryotic expression hosts like E. coli, non-pathogenic strains of Corynebacterium, Pseudomonas fluorescens, etc. And eukaryotic expression hosts include Saccharomyces cerevisiae, Pichia pastoris, Leishmaniella species, mammalian systems, etc. And the second major component of in vivo protein expression system is expression vector. Unlike normal cloning vector, this expression vector carries a tag which codes for affinity tags like polyarginine, polyhistidine, which actually code for the N or C terminals of expressed protein, and these overhangings, polyadenine or polyarginine or polyhistidine overhangings can be used for the protein identification, detection, etc. Commonly used expression vectors are PRZ and PET series. There are some examples of protein expression using in vivo system. Liu and co-workers in 2010 expressed recombinant glycoprotein using Pichia pastoris as host and PGAP series expression vector. And in Khan and co-workers in 2012 expressed the recombinant code protein of cucumber mosaic virus using E. coli as host, PQE30 as vector. Until now we have discussed about in vivo or cell based protein expression system. Then now we can move to cell free protein synthesis or in vitro protein synthesis method, which mainly deals with mRNA translation and post translational modifications, which can mainly used in production of large molecular size protein. It can produce more protein up to 400 kilodalton molecular weight, and it can deal with protein foldings and post-translational modifications. Although cell-free protein synthesis method is highly specific and it produces high quality protein, the quantity of protein produced is very less through this method, and the field is less exploited by plant virologists nowadays. And in vitro method mainly overcomes the tedious cell culturing methods of in vivo method, as we have already discussed in our earlier slides, and it mainly deals with the folding and post translational modifications, and we can architect our own protein of interest by incorporating or deleting specific amino acid sequences, and we can also express cytolethal toxins. Some protein may be lethal to expression host, and which cannot be expressed in using in vivo method, but that kind of protein can be expressed in vitro method, and we can direct the evolution of protein using mutation and immediate selection. Coming to the summary, there exists two types of protein expression system, in vitro and in vivo. In vivo expression system involve expression host and expression vector. Cell based, cell free protein expression synthesis, it actually taking out the engine out of the body and operating, and both the method enables high purified protein, and later this protein can used for further analysis. Thank you.